Actually, good afternoon, everyone. 10 minutes in. Thank you so much for being here this afternoon for what I think is going to be an amazing lecture. And I just want to start by welcoming you to now what is the 7th annual Richard V. Lee Lectureship in Global Health. So I'm Jean Witowski-Wendy. I serve as Dean of the School of Public Health and Health Professions. And today we're delighted to have Dr. Baccarelli here to give us a talk on public health epigenetics. You're going to learn something if you don't know something about epigenetics yet. And his talk will look at new paths to precision medicine. Now, this lecture is dedicated to Richard V. Lee, who's a former faculty member at the University of Buffalo. And the School of Public Health and Health Professions established this lectureship in Dr. Lee's name in 2016. And it was supported by an endowment from a close friend of Dr. Lee's. Dr. Lee, for those of you who didn't have the opportunity to meet him, I suggest you go read about him, but he was an amazing man who had contributions to many fields and many areas. And it was not just the University of Buffalo. He had an impact on the greater Western New York region. He was not a native of Buffalo. He was born in Islip, New York. He attended schools in New Hampshire and even Scotland. And he received both his undergraduate and medical degrees from Yale University. While in medical school, he was very prolific. He won awards, including the Ferris Prize in Anatomy, the Winteritz Pathology Prize. He did his residency and postdoctoral training also at Yale New Haven Hospital. After completing a residency, he worked for the Indian Health Service in Montana before returning to Yale as an assistant professor of clinical medicine. In 1976, he was recruited to the University of Buffalo, where he stayed the remainder of his career. He served as the chief medical officer at the Buffalo Veterans Administration Hospital, and he had faculty appointments in now the Jacobs School of Medicine and Biomedical Science, including in the departments of medicine, obstetrics and gynecology, as well as pediatrics. He was also a named professor in our School of Public Health and Health Professions in the Department of Epidemiology and Environmental Health, and he also had an appointment in the College of Arts and Sciences in the Department of Anthropology. So you can tell just by the appointments that he had how vast his impact was. He was also head of the Department of Medicine at the Buffalo Women and Children's Hospital for many years. In 2007, he received the C.C. Barnes Award from the International Society of Obstetrical Medicine in recognition of his outstanding contributions to the field. He served on the editorial and review roles for many medical journals, including the Journal of the American Medical Association, American Journal of Medicine, Journal of Infectious Disease, Annals of Internal Medicine, Journal of Clinical Allergy and Immunology, Journal of Clinical Epidemiology, and the American Tropical Medicine and Hygiene Journal. In addition to his involvement with many American organizations, he held memberships in the International Society for Travel Medicine, the Royal Society of Medicine, the Explorers Club, and the Royal Geographic Society. He also served as medical director for a Buffalo-based company, Ecology and Environment, and was a medical consultant to both the Buffalo Zoo and the Bronx Zoo. Beyond his extensive clinical work, teaching, service at the University of Buffalo, he found many opportunities for medical outreach work, primarily in Southeast Asia and China. He played an essential role in our university's programs in global health outreach, and the Office of International Education relied on his expertise as a medical consultant and cultural liaison. He also served as a consultant to the World Health Organization. He cared deeply, He cared deeply about environmental pollutants and their impact on human health, especially among women, infants, and children. When he traveled, he always paid close attention to environmental issues in the local communities. He and Dr. Baccarelli surely would have had much to talk about, and we enjoyed dinner together discussing with some of his friends and Dick's wife, Susan, discussing Dick and the many adventures he had taken. Dick was a clinician. He was a teacher, but he was also a humanitarian. His work impacted our community and the world. He drew upon his inexhaustible resource of knowledge and wonderful stories to capture his students' interests. He was a lifelong learner. He had ability to relate to anyone he met, regardless of culture, age, or station. He loved people and was devoted to helping them. Dr. Lee passed away in May 2013. He's sorely missed by all of us at UB and the greater Western New York region. We're so pleased, however, to honor him today with this lecture, and we're pleased that his wife Susan and his many friends could join us today for this celebration. Let me welcome all the faculty, students, community leaders, and partners, and UB alumnus that have joined us today. I hope you'll enjoy this lecture. I'd like to now introduce Dr. Lena Mew. Oh, you're way down there, Lena. Associate Professor in the Department of Epidemiology and Environmental Health, and also the Director of the School Public Health Profession Office of Global Health Initiatives, and she will be introducing Dr. Baccarelli.
Thank you very much, Jean, for the introduction, and good afternoon, everyone. I want to welcome you all to the 7th Annual Richard Lee Lectureship in Global Health. Today is really my great honor to introduce our guest speaker, Dr. Andrea Baccarelli. Dr. Baccarelli is the Liam Hans Professor and the Chair of the Department of Environmental Health Science at Columbia University Mailman School of Public Health. He also served as the director of the NIH-NIEHS P30 Center for Environmental Health in northern Manhattan. And his research investigates the molecular mechanisms as a pathway linking environmental exposure to human diseases. His current project investigates a range of the mechanisms, including epigenomics, the topic he will deliver today, and epitranscriptomics, extracellular vesicles, small non-coding RNAs, mitochondrial DNA, and microbiome. Dr. Baccarelli has really supported international best practice for air pollution control by multiple agencies worldwide, including United States, European countries, and many of the Asian countries. His finding has served as a basis for the Environmental Protection Agency's decision to enforce the stricter guideline for human exposure. In 2020, Dr. Baccarelli was elected to the National Academy of Medicine for his pioneering work showing that environmental exposure adversely affects the human epigenome. So far, he has published more than 570 peer-reviewed journal articles. And Dr. Baccarelli was featured in the 2020 Web of Science list of highly cited world's most influenced scientists of the past decade. Currently, he also serves as the president of International Society for Environmental Epidemiology. Now, please join me welcoming our guest speaker, Dr. Andrea Baccarelli. Thank you.
Thank you so much. Can you hear me now from here? Thank you so much for the very kind introduction and visit. We had an amazing night yesterday hearing about incredible stories from Charles and Dr. Lee's wife Susan about and his friends about his stories. And honestly, when I saw the list of speakers you had here in the past 6 editions, I got a little intimidated because you had amazing people who work at the White House, incredible. But after hearing about the life and examples that Dr. Lee did, I really felt very small. So thank you so much. It's a great honor to be here. It's really incredible to be here to honor Dr. Lee and his legacy. And it's really incredible to be able to present my science to you today. So let me start by telling you what I'm going to talk about. So you might expect now that I will tell you everything about the genes and how the genome is incredibly important. And I'll do that, but at first I would like to surprise you and start by saying that what we really need to worry about are not the genes, but the environment. In fact, I mean, if we look at what are the causes of human disease, you might find different estimates, but some very respectable estimates put the number that 70 to 90% of diseases are attributable to environmental causes, which many of those can be modified, which is, I think, part of the reason why we are in public health, because we think we can change this. So this is particularly important. And particularly, I want to start by using a term that many of you certainly are familiar with, which is the idea of the exposome. So we hear about the genome, the totality of the genes we have in our cells, but our field has started to talk about the totality of exposure. So the exposome is about the totality of exposures of any type, whether it's smoking, diet, social stress, environmental pollutants, and so on, is the totality that each of us experiences over our own lifetime. And I want to particularly emphasize the lifetime idea. We are not just happy to hear about the exposure we have today, but everything since we were born, and perhaps even before. So my interest, though, is much more on environmental chemicals and environmental pollutants. And the reason why we worry about that is because, indeed, we live in a chemical world. There are 3,000 chemicals introduced in the United States just every year. And if we look at all the chemicals that get either produced or imported in the United States, the number is in the trillions. So it's about 10 trillion pounds of chemicals that get introduced in the environment every year. And if you divide those by the 300 million of us who live in this country, it's about 30,000 pounds per person, which is honestly staggering. I mean, when I saw these numbers, I thought, this can't be possible. These are numbers from the EPA, from the Environmental Protection Agency. So the issue is we have a challenge here, and I want to speak about how we address that in my lab. So what we are doing in my lab is to use molecular biology and data science together to fingerprint this chemical exposure. So the idea is that, just seeing your reactions, we are unaware that we are exposed to all of these chemicals. So is there a way to use laboratory science to tell each of us what are the chemicals we are exposed to, and to be honest, what are the exposures we are exposed to, not just chemicals, and help us to protect ourselves against these threats. So there are a lot of challenges in doing that, and I will present 2 challenges and how we tackle them in my lab. The first challenge, first of all, let me go by saying that environmental exposures leave many types of fingerprints on human cells. So we are exposed to many different types of exposure, whether are chemicals, whether are bad nutrition, low physical activity, anything that can influence our body and our health that is outside of us. All of this changes our cells. So we published a review last year in Cell where we identified 8 domains of molecules inside our cells that can be used to fingerprint our exposure. I'm using the idea of fingerprinting, the idea that there is a unique fingerprint that each exposure might leave in our cells. And this, there are a few of them, DNA damage or mitochondrial dysfunction. The endocrine system clearly reacts to the environment and changes as we are exposed to different chemicals, intercellular communication, and so on. And you'll hear a lot from me about epigenetics. So let me start with the first challenge we want to tackle, which is time. You heard me saying we are interested in the exposome, all the chemicals all over our lifetime, but I mean, many of us are unaware about the exposure we have today, let alone over the past 10, 20, 40 years. So how do we tackle that? The good news is that we have something we can use, which is epigenetics. So epigenetics is about the changes in the DNA that do not depend in changes in the DNA sequence. And I'll go into this in a second, and I'll show you an example that I hope clarifies to everyone what it is and why I'm interested in it. But what is really interesting is that the epigenome is a way through which our cells build memories. So it creates memories in the cell that help our cells to react to the environment. And not just to react now, but these memories are stored into our chromosomes so that in 10 years, if we're exposed today to air pollution or dioxin, in 10 years, the body still remembers how to operate in those situations. It's like a standard operating protocol that once a threat is experienced, gets built in our cells, and in 10 years can be deployed again. And not only it can be deployed in our bodies, but there is evidence that this type of protocols can be transmitted to our children. So potentially we might have the opportunity that if there is a threat today, our children will be ready to go. And this is evident in plants. Plants built in memories based on the climate where they live. If they are moved to a different climate, they will reprogram their cells, they will build new memories. They get transmitted to future generations of plants. So the plants are moved from Florida to Buffalo. The future generation will know that the time of flowering, the rate of growth, needs to be on Buffalo climate, not on Florida climate. This is particularly important. It's an amazing way through our, to be honest, I'm always impressed how our bodies and our cells do so much that we don't have to worry about. Just think about how often we breathe without thinking that we are breathing, and that happens without any intention on our side. And our cells do exactly a lot more. So let me, though, stop for a second and give you an example to try to illustrate all of this. It's an analogy that is a musical analogy. And in my analogy, what you see here, the musical score, is the DNA. And you can understand how a musical score can be the DNA because the DNA is a code that is written in molecules, not in print, that can be used to create what we call a phenotype, which in this case is this You can go wherever in the world. I think this must be a music hall in Europe that where you are able to enjoy a symphony or any type of composition that you might want to enjoy. And that happens with the DNA. The DNA codes for all of our proteins, and that code creates our bodies. And what is really important is that you can think in music or in genetics, the DNA is not everything. You can give that score to me, and you can be sure you will not going to enjoy music as much as you were there, or just bring me there and get the director step down and even direct a wonderful orchestra. It won't be as good either. But so, I mean, this also it already tells you my point. Genetics is not everything. It's all about execution, correct? And this is something that in genomics we know as incomplete penetrance or genetic or phenotypic variation. It's something that we know very well. People with the same genes, think about twins, experience different lives, have different risk of disease. And this is because the environment is very important. But you haven't yet seen the epigenome. So, in fact, if you want to see the epigenome, you can't stay on your seat over there, but you need to go on stage, look over the shoulders of each of the performers, and then probably you'll see something different. You will not just see the score as it is in print, but you will see a score where there are probably markings added onto the score. When people rehearse, often write down markings, and the conductor might say, let's do this passage this way, or they need to remember how to remind themselves how to do things in a certain way, and they would add marking onto the score. And this is what the epigenome is about. There are markings added onto the DNA that doesn't change the sequence, doesn't change the score, but it does change the way the DNA is performed. And particularly these marks help decide the cells whether the genes need to be on or off. We all have 20,000 genes in our cells, but each cell uses only 1,000 of them. And you and I don't have to worry about the billion cells in our body, which genes are going to be on and which genes are going to be off. The cells themselves decide, and the epigenome is one of the mechanisms through which cells activate and inactivate genes, and meaning whether genes produce proteins or not. And so this is really an incredibly sophisticated system, and it's all about these annotations that are added onto the score that decide that that specific gene is going to be off, or vice versa, this other gene is going to be on. And particularly what we learned over the years in studying epigenetics is that this type of marking can be classified in 3 types with a very scientific classification I created that classifies them in ink, in hard pencil, or soft pencil. And the idea is this, is that you have the DNA, and some of these markings, once they're applied, they're forever. They're never going to be erased. For instance, the epigenome is also very important to differentiate our cells. We are all come from a single cell, the zygote. We become a small embryo, and the zygote and the morula and the early stages of embryo life, cells are very similar, and then they start to pick different lineages, and then they become brain cells, muscle cells, skin cells, and the muscle cells need to produce muscle genes, but not brain genes, and vice versa. And this is driven by epigenetics. And those marks are very, very hard to erase. They basically never get erased. So they are the one in ink. And we found that there are some environmental cues that can add these ink marks that then they stick around together. But then there are marks that are in pencil. For instance, the immune system. We have inflammation is one of the defenses of the immune system that need to be deployed stat. If we have a virus in our body, the immune system needs to act now, not in 10 years. So there are some of these genes that are very, very, very fast, like in a matter of minutes. So these are the ones that I call soft pencil. So but now I think it's time to get out of the analogy and tell you about what is this really is. So what we study in my lab is one of the epigenetic mechanisms, not the only one, is DNA methylation. DNA methylation is a very simple chemical modification of cytosine. You see up here, if I can point to this, these are cytosine followed by a G, so it's we call it a CG dinucleotide. The DNA has 4 bases, and the one that gets modified in humans is a C, is a cytosine. It's a very simple chemical modification. For those of you who have a chemistry background, it's a methyl group, a CH3, 1 carbon and 3 hydrogens added onto the DNA. And when a gene is methylated, usually we believe is off, while if it is unmethylated, it's ready to be on, or it can be already on. So we say it's poised to be active. So this what happens, and cells take all these executive decisions about which set of 1,000 genes needs to be on, and a lot has to do with DNA methylation. And so I'm going to go a little bit into the technology we use. The way we measure DNA methylation in my lab is by using this technology that is called a microarray. This is a very small chip, not unlike the chip of a computer. It's something this big, like a couple of inches. Each of these chips houses 8 samples in our iteration, so we can put 8 DNA samples. And a lot of what we do, I probably was clear from the animation I had with the blood tubes, a lot of what we are trying to do is to use blood, not because it's the best tissue, but it's because the easiest to get. So we put 8 blood samples on there from 8 different people, and then we can measure, by doing that, we can measure 850,000 methylation sites. So we have 20,000 genes. That really means we are measuring around 40 methylation sites on each gene, in average. So it's really, when you send out a sample for methylation analysis, you get back 850,000 numbers. So we have a lot to deal with, and lots of my students and postdocs are really at this point data scientists. It's what we mostly do in my lab. Especially, I mean, it's interesting to see this because it's really a lot of data and a lot of information, but also it means that we have a powerful tool to fingerprint exposures. And for instance, I'm going to show you a little bit what we have been doing with this type of technology with DNA methylation. And I need to also say that the numbers we get back is a continuous measure bounded between 0 and 1. So it's a number that varies between 0 and 1 and can take any value. Where 1 is a sample where all cells are methylated at that site that we're studying, at one of those 850,000 sites, and 0 is a sample where all cells are not methylated. And then we can get a proportion of cells methylated between 0 and 1. It can be 2%, 20%, or 75%, depending on the samples. So using this technology and using blood, we try to see whether the environment does change the epigenome, and it does. For instance, this is a study we did in a consortium in Europe and the United States. This was 1,500, this was supposed to be mother-child pairs, not mother-blood pairs, sorry about that. So 1,500 and some mother-child pairs from 9 cohorts in the United States and Europe. And we did take cord blood. So cord blood comes from the child, from the newborn. It's blood that is circulating in the child. And we measured the DNA methylation in cord blood. And we found that the level of pollution, of air pollution, this particulate matter, PM2.5, for those who are familiar with air pollution measurements, the level of air pollution was associated with differences in methylation. So where you live during pregnancy seems to have the fetus react and to change the methylation status of certain genes to mostly cope with the levels of exposure. And just if you're interested to see what we do graphically, this is what we call a Manhattan plot, because it looks a little bit like the skyline in Manhattan. And it's the official name. This time I didn't make it up, uncharacteristically for me. So this is the chromosome number. We have 23 pairs of chromosomes. We don't use usually the X and Y chromosomes for mostly technical reasons. So we analyzed the chromosome 1 to 22, and this each data point here represents one of the 850,000 methylation sites. And the only thing we are plotting here is the statistical significance. So this is the inverse of the log 10 p-value. So the higher is the point, the taller is the point in the plot, the more statistical significance it is. And this is the threshold we use to test statistical significance. So the red diamonds are the ones that were statistically significant. And I can tell you a lot about which genes they were, but for today's purpose, I would just like to show this as an example that indeed we can detect, we can fingerprint exposures that are around us using this method. I would like to go more instead into the properties of these mechanisms. So something that was really impressive in a similar consortium, this is a consortium paper that was led by Dr. Johannes in Framingham and Dr. London in North Carolina at NIHS, and we were part of it as part of the consortium. So we contributed together 16,000 volunteers from different studies, from 16 studies. So we had about 16,000 blood DNA samples. We measured methylation with the same tools. And again, I mentioned that we had 20,000 genes, and we found that smokers had differences in DNA methylation compared to non-smokers in 7,000 of them. So this tells you how heavy smoking it is for our cells. Cells react dramatically to smoking to the point that reprogram and get ready 7,000 genes to cope with smoking. So that's about 40% of the human genome being modified. We never saw anything like this in any of the studies. This is the biggest difference I've ever seen in my life, and it's due to smoking. But what's really interesting is that we had a few people, quite a big number of people who quit. And when we looked at those who quit, we found that many of these 7,000 genes went back to non-smoker level in a few months, many in a few years, but there were a few, and I'm showing those here, that never went back to normal, never went back to non-smoking level. You see, these are years after people quit. So we had a few people who had quit for more than 30 years, and these genes were still different, including one that is very well known now in the epigenetic world, which is called the aryl hydrocarbon receptor repressor, that was lucky enough to be the gene I spent my postdoc life working on, then completely forgot it existed, and after 10 years it came back in this study. It's a gene that is known to be in a pathway that is very well known to be activated by smoking, and this repressor particularly gets activated because it makes the cells less vulnerable to smoking. So this is really interesting. So the question at this point is, we found these genes in ink. My interest is to use them almost as a time machine to go back in time and address the issue of time, address the issue that we want to measure what people did to their bodies, not just now, but in the past 10, 20, 30 years or more. And so, fortunately, some of our colleagues working in this area decided to take data sets like the one I show you with 7,000 signal modified and other similar data sets and use machine learning to identify what are the methylation sites that we can use to tell us whether people smoke or not, whether whether they, how long they've smoked, and potentially even the number of cigarettes they smoked in their lifetime. So there are these blood-based biomarkers that are basically a combination of methylation sites associated with smoking that you can use, and you can get someone's blood, measure the methylation, plug the data into these algorithms, and they will tell you whether someone is a smoker or not and how much they smoked. And potentially this is lifetime. It has the potential of being lifetime. And so it's really an interesting way of doing it. For those who are into machine learning, the type of tools we use is called ElasticNet. That just selects the best predictors of smoking, and so we can go back and see what what was the smoking status of the person we got the blood from. One of these tools is called epismokers, which is actually one of the simpler tools. It just classifies people in smokers and non-smokers or former smokers. And one of my team members used this to try to see whether we could use it to predict disease. And particularly, she's a pulmonologist, Christina Eckard up there, and she had the idea to look at blood DNA methylation at baseline today, and then see over time, over 10, 15 years, what was the situation in terms of lung disease. This is a study which is called the normative aging study. It's based in Boston, in the Boston area. They are veterans, pretty old at this point. They are in their 80s right now. And so many of them had chronic lung disease. And you can see how the blood-based biomarker, the methylation biomarker, predicted very strongly the difference between smokers. And so the smokers down here had much higher risk of lung disease than the non-smokers. And she also looked at mortality. This is all-cause mortality. And unfortunately, as expected, the people who smoked had much higher mortality than the people who didn't smoke. This was expected. I mean, we know that smoking is bad. I have friends who periodically meet me and say, "Oh, you are still studying pollution. Have you actually proven that pollution is bad?" And once in a while, we need to remember that we know that smoking is bad. But the situation here is really, what was really interesting is that this predictor, this methylation predictor, which is supposed to predict only whether people smoke or not, was a much stronger predictor of lung disease than smoking itself. So the self-reported smoking didn't seem to be associated so strongly as this predictor. This is perhaps because this is built in our cells, so it records more than just our memory of smoking. It might be more accurate because cells don't lie, people can. But at the same time, it might just be that it's within the cells. So it also measures whether smoke and products of tobacco smoking get into the cells. So this is what we had, and we went one step further. One of my postdocs, who's now an assistant professor at Mount Sinai in New York, Elena Colicino, had the idea of see whether we could create a similar predictor based on DNA methylation in blood for lead. I don't have to explain how lead is important. It's a toxic metal that has been associated with many neurological and cardiovascular adverse effects. And she had the idea of using the same machine learning approach to look at whether she could create a DNA methylation-based biomarker. And we were very lucky because we had a unique database where we had measurements of lead in bone. Lead is usually measured in blood. And it's measured in the entire blood, and what we measure in blood as clinicians is something that is worth about 90 to 100, 3 to 6 months of time, so it can reflect what we were exposed to in the past 3 to 6 months. Bone is interesting because lead accumulates in bone, it sticks into the bone, and half-life in bones is about 10 years. So when you measure bone lead, you had 10 years worth of exposure. And just before you accuse me to be a criminal, we didn't take bone biopsies. These are 700 people. There are 2 centers in the world, I believe, that can use X-rays. It's a very sophisticated X-ray equipment to measure lead, in bones. And so we had these unique measurements, and Elena was able to replicate what you saw for smoking and create a methylation-based biomarker that predicts bone lead. And what was really surprising is that the biomarker didn't seem to work extremely well. These are the real bone levels of lead. These are the estimated methylation-based. And you can see the correlation is there, is significant, but it's not that great. These are cross-validation for those of you who are in machine learning. But in fact, this seems to appear, it was a blessing in disguise. When we use this biomarker to look at disease, we found that this biomarker, the methylation-based, is much better at predicting cardiovascular disease than lead itself, in the same way as I explained for smoking. So this is where we are with this type of measurements. But I want to pause a second and dream about the future. So what we are working to, my vision is that in the future, you would have this so much evolved, so much well established that you will go in your doctor's office every year if you go, and they would, in addition to, of course, annoying you with your cholesterol and eating less eggs and meat and so on, and measuring your glucose and so on, they will also measure methylation, and they will measure everything we spoke about. And potentially, I even decided to spend a little bit of my time since I decided it was more fun than doing the things that my dean wants me to do. Sorry, Jean. And made a possible chart of what in 10, 20, 30 years this will look like. And so that you would think about, you will go to your doctor's office and they will tell you, you are doing great with smoking, and you probably know because you don't smoke. But, we found that there might be something in your water or food about metals. So we probably need to test your water because, we saw that the metal genes have moved up. There is something going on with your metals. Or perhaps your air pollution. Or you can think about, you probably work out 200 days a week, and you're doing well with physical activity, but, this is really autobiographical. My doctor always tells me I shouldn't be drinking at all. He's convinced that alcohol is bad in any quantity, so I decided my 1 glass of wine might be increasing my risk, though probably it's not true. Don't take that. As an Italian, I'll always believe it's not true, even if there is great science. There is no great science, but if there were, I wouldn't believe it anyhow. and so this is really something that is really interesting. And potentially you could think that you would also have a dial that reflects your risk of disease. And because I spoke about lung disease, I really decided to say, all of this is going to cause your lungs to age a little faster, and perhaps you are more prone to lung disease. Let's do something about this. And I think there is great potential to help people to modify their lifestyles, to modify their exposures, and to identify problems that they're not aware of, and to help them fix them. So my idea is that there will be an environmental diagnostics in the future, like we have diagnostics for a lot of different areas, and some of them are environmental. In the end, cholesterol and triglycerides are also environmental, though of course there are also genetic components that we will have in the future. So this is where I am. Please take a picture, so if in 30 years I'm right, I'll be very happy to having been right all along. If I'm not, we had a great ride. And this is why I think using blood DNA, we were discussing today with some of our colleagues, it may be very important because blood is easy to get. Though you could do this in saliva, potentially, like 23andMe does, and so on. So I would like at this point talk about a second part of my presentation, and the second challenge and the second technology. And it is about tissues. I mean, I spoke about how we are exposed to lead, how we are exposed to smoking, how we are exposed to air pollution. And I mean, oftentimes when we study these threats, we don't take into account how complex our body is. We have many tissues, they're all different. We are measuring methylation in blood, but blood doesn't reflect anything of what actually happens in the tissues except blood. And if you go on Wikipedia, which has become my to-go source for scientific understanding, you get, if you ask Siri how many cell types there are in the human body, it will send you to Wikipedia where someone wrote there are 200 cell types in the human body. And I think it's an underestimate, depending how you really identify cell types. I spoke with an immunologist who thinks there are at least different 300 cell types only in white blood cells. So, I mean, depending how you think what a cell type is, you might have a different answer. But let's say there are 200, and we are taking a single measure in blood. How do we interrogate potentially each of these 200 cell types? And this is a great challenge because disease often is tissue-specific, cell type-specific, organ-specific. And to make a case about this, I'll go to our research on air pollution and dementia. As you know, this is a huge issue worldwide, but we recently published a paper together with my colleague Xu Gao, who's now an assistant professor at Peking University. He used to be in my lab as a postdoc, where we showed that air pollution is related to cognition, and we found both long-term effects. I mean, air pollution today is associated with future risk of dementia over the years, but also fluctuations in air pollution seem to impact cognitive capacity in older people. Bad pollution days are also bad cognition days, apparently. And so this got worldwide coverage, and at this point we are convinced that air pollution somehow gets into the brain. Some of the particles and the gases can go directly into the brain and damage the neurons. Some others probably activate inflammation in the lungs, and inflammation does the damage everywhere else. But I mean, the question is, how do we detect this type of damage? The damage is in the brain, and unfortunately in the study, when I spoke with people who do animal experiments, they say, oh, it's easy, we'll sacrifice the rat, the mouse will get a piece of brain. Of course, I mean, in humans, you can't get pieces of brain of people who are alive. I mean, there are people who donate their brains to science after their death, but we are really interested in something else. What we are interested in specifically is this idea. The bad news for some of us is that Alzheimer's doesn't start when you're old. It starts when you are very young. So some of these people who have donated brain to science have enabled us to do studies where we can look at the brains and see whether we find signs of Alzheimer's in people's brain. And scientists have found signs of Alzheimer's in brains very early in life. We are talking about the early 20s or 30s. So down here, like 30 or 40 years before Alzheimer's actually show, you already find brains that have Alzheimer's damage, many of them. And but the symptoms usually start to appear down here, and usually Alzheimer's, 99% of cases of Alzheimer's are diagnosed in people 70 or older. So really, you have a disease that is down here, but it starts up here. So the question is, how can we access the brain in people who want to live the next 40 years, not in people who already died, to understand what is the situation, whether we can find the damage in there. So we also have a possible solution here, which is extracellular vesicles. So these are small membrane-bound vesicles. You can see there are those small things over here and over here. They are secreted by all human cells. All of our cells in the body release these type of vesicles. They're membrane-bound, they're small, they're little bubbles, and they're found in every human biofluid, including the bloodstream, including plasma or serum, and they're known to be like a previously hidden signaling system. Particularly, we are interested in the microRNAs. I will talk a second what they are. Actually, I can do that now. So there is plenty of microRNA in extracellular vesicles. These are very small RNAs. For those of you who are not in biology, the DNA produces RNA, the RNA produces proteins. So the RNA is really in the middle between the genes and the disease and the phenotype. But these are very small RNA that do not produce proteins, but they can bind the messenger RNAs, which are the ones that produce protein. And they really, I know that we are in a football town, they really work like a linebacker or a cornerback tackling the running back or the opposing player. They really tackle the messenger RNA and don't let it go. And the messenger RNA at that point cannot produce proteins. So the net effect is that they shut down genes at the post-transcriptional level. And we always studied this as being inside a cell. So a cell produces the messenger RNA, but also produces the micro, and they have this balance where the messenger can be tackled by the micro. But now we understood that these microRNAs are inside the vesicles. So what we think might be happening is something like an email system or a signaling system, using a scientific term. But using a more lay people term, is like our cells are sending emails to each other. So you can imagine about, here again, this is something that actually one of my students came up with this, Rhodosthenis. So the idea is that you have the lung epithelial cells who send an email to the neutrophils, which are in the blood, and to the cells around them, the lung alveolar cells, and perhaps even the brain, saying, you know, there is a problem, we have air pollution here, and say, dear neutrophils, as you are fully aware, over the next few days, we have been exposed to air pollution that is higher than usual. We really need your help here. Please give me your attention. And there will be, please see attachment. And the attachment will be microRNA, including vesicles, that go to the neutrophils and tell the neutrophils what to do, tell which genes need to be activated in this situation, which genes instead they're okay. And so there is this really, we also made up an email that of course doesn't work. Don't expect the epithelial cells to answer to your email. You can try, but the idea is really that there is a communication. In a way, I mean, cells can communicate with each other in the same way we do as humans by email. But the signal, the real message is these microRNAs. There are hundreds of microRNAs that can tell the recipient cell, the neutrophils or the brain, which genes need to be shut down in this situation. So, of course, I mean, we are again talking about time. Before we talked about the past, now we are talking about the future. If we want to predict Alzheimer's, we better have a longitudinal study, because we want to predict Alzheimer's in the future, not in the past. And so we used a study that I already mentioned before for the bone-led study, which is the normative aging study. This was a large study started in the Boston area in the VA hospital in the 60s. There were more than 4,000 people that used to be followed up, and in 1995 there were still about 650 that were active, but they were still coming back. And especially we had plasma blood stored for all of them. They come back every 3 to 5 years. They still come back, some of them. And we found that we isolated the vesicles from plasma, and we found that there were 70%, there were about 400 of these small microRNAs that were found in at least 70% of the plasma samples. And we also, these people also have an assessment of cognition, something that is called the mini-mental state examination. There are 30 questions about how they measure different domains. Mostly whether they remember their zip code, whether they know who's the president of the United States, and whether, and particularly in Boston, there is one question we had to throw out, which was, what is the name of your county? Apparently, Bostonians don't remember their county, no matter what. Doesn't depend on, counties are never used for any type of official purpose, so people don't really know which county they live on. And the mini-mental examination became very famous 2 years ago, I think, or 3, when President Trump was administered one. You may remember that this really, he boasted that he did well, but this is a test to screen for dementia. So if I were him, I wouldn't be so proud that my doctor actually thought I need a test for dementia. But I mean, this is really what we did. It's 30 questions, a score that goes to 0 to 30, where 30 you are great. In this case, 29, because one of the questions couldn't be used. So we measured the microRNAs in plasma at baseline, and this is work done by my former PhD student, Nicole Comfort. And she found a lot of microRNAs associated with future cognitive decline. So people who went down with their scores were the ones who had these microRNAs either increased or decreased. So this is another plot that's very similar to the Manhattan plot. The field calls this volcano plot. You can imagine why, very descriptive. On this side is the direction of the association. So these are each dot is a microRNA, and these are the positive associations between microRNA and cognition. So if the microRNA is high, these people are going to do great, and vice versa. And this is the p-value, the inverse of the p-value. So again, these are the ones that are significant. So she found very many, really a lot, and we are trying now to combine them using machine learning to see whether we can create an algorithm again, and also found a lot of different pathways that these microRNAs belong to, including very many that are important for cognition and brain functioning. So I have to say, you might have noticed that I started with the promise of speaking about how we can interrogate different tissues, and this is not yet that. This is just to convince you that vesicles are important and that contain message that may predict cognitive decline. But here we measure all the vesicles together. By the way, we had data showing that some of these pathways are also activated by air pollution, and we are trying to put everything together now. But so how do we get to the individual tissues? The good news is that we can isolate vesicles released by neurons. So the vesicles have on their surface proteins that come from the cell of origin. In this case, neurons, but it can be any cell in the human body. So we can identify vesicles based on where they come from if we know the protein. So we use an antibody to pull down the proteins, to immobilize the protein, pull down the vesicle, and then we can isolate only the vesicles that come from neurons. These vesicles cross the blood-brain barrier freely, so we are hopeful that many of those will come from the brain. And then we can isolate the RNA and sequence only the RNA that come from neurons. This is work done by Howie Wu, who's a postdoc in my lab, and he was able, for instance, to show that he can enrich the vesicles for oligodendrocytes. It become 100-fold enrichment. We didn't do well in the beginning with neurons and microglia, but the latest data he showed me, we had to change the antibody, and now the neurons are up here as well. So we can really amplify the signal 100-fold and interrogate specifically the neurons, or the microglia or the oligodendrocytes, or even down the line, This is the grand plan we have. We want to develop an atlas with 20 plus cell types that we can interrogate that come from all the tissues associated with aging and more. And Diane Reyes, an assistant professor in our department, also showed that she can measure chemicals in these vesicles. So for instance, she has been doing work in neuron-derived vesicles where she measures metals in vesicles that come from neurons, and she demonstrated that they are a great biomarker of brain metal load, or the load of metals in the brain, which is something we never had. We have never been able to predict, to measure exposures in a certain organ, and now we can. So this is really what cancer scientists would call a liquid biopsy. It's a way to biopsy the brain without having to take a piece of the brain, which I wouldn't want to do, have done on me at least. So, of course, there are challenges. We have a lot of exposures to investigate. I gave you the example of smoking, of lead, metals for vesicles, but these are just a few. Many others are working on other exposures, and hopefully there will be a lot coming forward in the future. Particularly what is challenging is that if you want to create lifetime biomarkers, you need to train the biomarker on lifetime data. Someone has to give you the data about that exposure. So this is why smoking is great, because you can ask people to recall approximately their smoking history, and that helps you. But we really need longitudinal studies to do that for other exposures. And of course, these biomarkers need to be validated across different populations. So this is really a little bit of the science ahead. I want to, at this point, thank all of my lab team and collaborators. You saw a lot of the junior people in my slides, but there are a lot of senior people who also helped me a lot. We are incredibly lucky to be funded currently by the grants you see here from NIHS, NIH Aging, and other institutes, including our center, P30 Center. And I would like also to tell you that we really believe that data science is important for public health and environmental health. I'm sure I don't need to convince you all here. And we are launching now a Master of Science in Data Science for Environmental Health. So if you know students who want to work in environmental health and love data, send them our way. Also, if you are interested in learning more about what I talked today, we have this hybrid program. Some of the boot camps are online, some are not. These are very short boot camps that we offer out of my department. That, for instance, I teach together with my colleagues an epigenetic boot camp, which is 2 days, and it's going to be online in the summer. If you want to learn more about epigenetics, look us up. Or if you are interested in other different areas like environmental mixtures or the exposome or machine learning, you can look us up as well. And many of those are online, some are in person. So this is all I had, I think, and I hope I left some time for questions. And thank you so much for having me here.
Thank you so much, Dr. Baccarelli, for such a stimulating lecture. It's really fascinating. And now we have time for some questions. Yes, Laurie?
So I'm standing up so people can hear me. Oh, good.
So that was an amazing talk. I really learned a lot. And one of my concerns is the focus on the individual rather than the focus on the broader environment. I mean, when you talk about air pollution, for example, as an individual, there's not a whole lot I can do. And we do know that there are communities that are inundated with air pollution. I know Lena did some very interesting research when, due to the Olympics, they shut down industries in Beijing, and then when they, everything kind of calmed down and people had positive experiences with breathing, and then when it was over, everything's back to non-functioning or very difficult situations around breathing. So have you and your colleagues given any thought to how you could move some of this into the policy arena? Because even though I'm not a smoker, I do breathe, and so I could get affected by air pollution.
Absolutely. I think there are two answers that I can offer to your question. You're completely right. I mean, my presentation focuses on the individuals with a model on precision medicine, but you could present the same data at the population level. I think there are two types of answers. First is that if this type of technology becomes widely available, it's already very easy to scale it up to populations, and you can monitor populations very easily, perhaps more easily than what you can do today with air monitors. And honestly, the real interest in terms of monitoring is that it's one test, one blood-based test that potentially can measure hundreds of risks. So potentially in terms of looking at different threats in the population, this would be an amazing tool. If we have an array of 100 or 1,000 different biomarkers that we can use to measure air pollution, social stressors. I mean, we have evidence, for instance, that women who are exposed to, who suffer from abuse during childhood have permanent changes in DNA methylation that last forever. So even screening people for that would be incredible. So one answer is that, and this type of technology now costs around $150 per sample, but the real cost is around 10 cents. So at the day this can be blown up, the cost is going to go down, especially the day that the Illumina loses their patent, because that is really what's keeping it up. And so really having something that potentially is cheap and easy would be amazing. The other thing is that you heard from Lina that some of my findings were used by different countries in regulating air pollution standards. And they were basically these finding that air pollution impacts the epigenome. And I have to say, I'm not sure I agree with the thinking, but the DNA has such a magic place in our imagination, in our culture. I mean, in a way, it's almost like, I was in Greece 2 months ago, and I visited the Parthenon and all the gods and goddesses of Greek. I think the DNA is about the same space in our culture, because, I mean, we really think the DNA is the dominator of our life. So, I mean, when people saw, I have reports in my computer from countries and cities all over the world that they saw our papers, and the regulators were start to be convinced that air pollution is bad. They didn't care that people die, because, you know, people die anyhow, apparently. Apparently people dying is not important. But when you see that air pollution gets into your DNA, people are completely freaked out. So in a way, I think it's also important to raise awareness about these issues. I think just the fact that we know that it's not, in reality, I mean, stopping joking for a second, I mean, in reality, it's important to know that our cells react to air pollution. And I think that has been used also to motivate regulations. But I fully agree, I mean, there is no way primary prevention in environmental health is removing the exposure. There is no other way to operate, to do that and removing the exposure for everyone, not removing the exposure individually. On the counter, unfortunately, we know that air pollution is not going to go away. I mean, there is always going to be a little level of air pollution, because some of that is natural. It comes from the sea salt, it comes from the ground, from the minerals. So even that can cause health effects. So in a way, even if we created a world where there is no human emission, there would still be some air pollution. So I think this might be helpful also in a world where humans don't produce any air pollution.
Well, thank you for all that you're doing, because I think it's really important.
Thank you.
How you doing? My question kind of encompasses a lot of what you talked about in terms of major challenges, from lots of pollutant, time, and tissue. And so I'm wondering, is there models that can be used for dynamic changes in methylation or any other kind of epigenetic modifications, such as potentially like organoid models, cerebral organoid, like you mentioned, a dimension study, lung organoid models, where you can do more of like a longitudinal kind of high throughput toxin screening in a dish.
That would be amazing. Never thought about this. If you are interested, if you're looking for opportunities to do that, I'm happy to think about that. But I haven't thought about that with DNA methylation, but it's exactly what we are funded to do for the extracellular vesicles. So the way we want to demonstrate that cells send email to each other is exactly by doing in vitro studies, and we are planning to use brain organoids. So we want to get lung cells, put them in a petri dish, dump pollution on it, in particularly particles from the air pollution, and then we are going to harvest the signals that come out, the vesicles, from the supernatant, and move them in another petri dish where there will be a brain organoid, and see how the brain organoid reacts to that. And of course, I mean, we can do other things like mimicking, creating synthetic signals that are similar to the one we see in vesicles and putting them directly on the brain organoid, or using other RNA that stops the microRNAs in the vesicle. It's called antisense RNA. And this is really interesting because I didn't mention this, but the non-coding RNAs are very interesting for treatments, for drug development. There is a lot of work in bioengineering to create vesicles that have specific signals to go into tissues and change the gene expression in the recipient tissue. So potentially that could be not just a fun experiment, but also with lots of applications in medicine.
Other questions?
Thank you very much for a really great presentation. And so we've been looking at these markers for deleterious effects. And there was this really silly movie that I saw a few years ago where they were going to send an astronaut to Saturn. And they decided that they were going to modify his DNA so that he would be better able to survive in that hostile environment. And actually turned him into a grotesque sort of thing, right? But I was just wondering, like, could we be using the same thing to enhance certain attributes of people? You know, if you wanted to be a better piano player, for example, can you do something beyond, you know, practicing more, or, you know, pick a skill, pick a whatever? Does it work that way?
I mean, in principle, we can certainly do things like that. I think at this moment there is a lot of work on gene editing, and so we can. The real interest, I mean, I started to train in medicine when we were not. Thinking about measuring the entire genome was a dream. The sequence, the Human Genome Project was a dream, and in 5-10 years, everything came to place, and we were able to sequence an entire human genome. And now we can sequence any lab in the world that can sequence a human genome. But the real excitement now is that we can rewrite the genome. So there is gene editing is a reality. And there is a lot in that. I understand that it's being used a lot to treat diseases that are single gene disease, like cystic fibrosis or similar diseases. And honestly, those applications are amazing. Being able to cure a genetic disease is something that we never thought would be possible. While today we can cure genetic diseases. And the innovation also in the epigenome is that now we have similar techniques to rewrite the epigenome. It's much less advanced, and we still need to learn a lot, but it will help us a lot to learn how the epigenome works, because now we can really control the epigenome in vitro or in animals, and see how cells react when we try to switch a gene on and off, which is something we couldn't do even 5 years ago. So, I mean, there is a lot going on. Actually, I came back from the annual meeting of the National Academy of Medicine, and there was a similar scenario. A colleague from Stanford who's working on optogenetics was telling how we can change the brains change the functioning of brains in rats, and that changes completely the perception of reality. So rats perceive reality, an environment that seems hostile and creates a fear reaction which is a model of post-traumatic stress disorder, they can engineer neurons that change completely the perception of rats. I was saying it would be great to treat mental health, but at the same time we need to be very cautious about what we do, because there is an incredible variation on neurophenotypes in humans. That is good. We don't want to create a normal human who's only a certain type of human. That would be terrible. So it really, but at the same time, these technologies exist and they work in animals, so we have no reason to think wouldn't work in humans.
Yeah, one more question.
Hi, thank you very much for this lecture. It was really great. So my question is, can you hear me?
Yeah, a little louder, please.
Okay.
Thank you.
Yeah. So my question is like, does saliva contain the extracellular vesicles, and if so, we can collect DNA and extracellular vesicles from saliva, then can we do all the experiments that you did with the blood instead of doing the blood with the saliva?
So, I mean, you can definitely do exactly what I do. The question would be, where do the saliva EVs come from? So the great thing about blood is that all cells dump vesicles into blood, while in saliva probably you would have only vesicles that come from the mouth. Though, I mean, I'm not completely sure. I mean, there is a lot of, I haven't worked in saliva as much. We actually have a study on saliva, but we haven't worked about looking where the cells come from. But I would think most of them come from the salivary gland and the lining of the mouth and the inflammatory cells that are in the mouth. But I might be wrong. They might be coming from everywhere as well. So it would be interesting to see. Actually, probably doing some studies trying to figure out where they come from would be interesting as a first step. So that's the only limitation I see. But yes, you can use saliva, absolutely.